Okay, I'm gonna explain to you some of the basics. I'll probably make several of these videos because it's easier for me than writing it all out. Some of the basics of what we're doing and also kind of why there's a delay in me sending you the data. It has to do with file sizes and me figuring out the best way to get you to do the most work I can. So let's get started. And I don't have a tripod, so this is held up by Red Bull and tape. So if it falls, chug a cup of coffee. It'll be a game. Okay, and hopefully this won't start dancing. You'll know what that means if it happens. So basically this is a program I use for mapping. When you get the files off of the sequencer from the sequencing facility, they're in something called a FASTQ format. And these are very large files of all of the data off the machine. The first thing you do is quality you trim the ends and then you filter for quality. So I've already done that and I'm not going to have you redo that step. But the next thing we do is we map, and I am going to have you do some of that. I already have all that done for my stuff. And I'm going to show you what that looks like. If I can. So this is chromosome 7. This is all of the chromosome. Everywhere you see a green or red or yellow, all of that, those are called annotations. That's basically what's at that gene. So this is, or what's at that location on the DNA. This is DNA. These are our RNA sequences, I believe. So this is called FAM20C gene. This is all of the genes, and all of these black lines are reads that map. So in the FASTQ file, you basically have lines of data, short sequences of RNA data, and those are each called a read. So one sequence is one read, R-E-A-D. So these are all the reads that map, and you can see a lot of reads map here. As we zoom in, which my mouse button isn't scrolling. Let me pick up this gene. So this is the gene we were looking at. You won't be using this program, but this is a really nice program. You have to pay to use it. It's done by some people in Australia or New Zealand, and they've built a really nice program. So you can visually see the sequences. And basically what we have is this is a read, one read, and it maps to this, which is the reference. Where it's colored, that's where the base disagrees. Now here we don't know why the base disagrees. It could be an error in sequencing, or it could be a phenotype difference from the reference genome. At this point, the reference genome for humans is built from many people and data sets, versus sometimes if you're working with an animal, it'll be built from one specific animal, in which case you might just have an allelic or just a nucleotide difference in the subject you're sequencing versus in the animal that you made the reference genome. So make sure you understand whatever keywords, whatever you've never heard before, write it down. So reference genome, reads, these are the IDs that come off the FASTQ. You don't have to really worry about them, but this is each individual read, and this is all for one person right here. So here, where there are some different nucleotides, because it's on the end of the sequence, it's probably just something left over. We call it a sequencing artifact, and it's something left over from the lab processing of the material. So in order to sequence, we have to add extra nucleotides onto the either end of the read, and sometimes that doesn't get completely trimmed off. One of the reasons why I like Genius mapping is it doesn't really matter if some of this is left on, because you can see that it still maps. Okay, now the basics of mapping are, with Genius, it uses a K-mer search, so K-MER, K-mer. That would be like oligomer, nonomer would be 9, a length of 9 nucleotides. The K-mer search, basically, it pulls out a certain length of sequence and it searches this length against all of the reads. Once it finds reads that match, it goes out one nucleotide this way, one nucleotide this way. I can't remember where I pulled from. But anyways, it just keeps kind of extending. And as it extends and more matches, then it assigns this read to this section of the genome. Now, when this is done, importantly, all 25 chromosomes are used. So 25 for male. I actually leave the Y chromosome out initially. I'm not going to get into that right now, but the 25 chromosomes are 1 through 23. 1 through 22, sorry. 1 through 22 somatic chromosomes, and then you have X, you have Y, and then you have mitochondria. So that's why I say 25. And initially this is mapped to everything but the Y chromosome, so it's mapped to 24 chromosomes, even though we're just looking at chromosome 7. So if there's a disagreement, like if this section right here maps across multiple genes, then it has to be assigned to one gene or the other. That's kind of the basics of the mapping. It just searches for a specific smaller sequence and then it extends out to see what else matches. Yeah, I won't get into other stuff right now. So I already have this all done, and this has most genes and most microRNA, but it's from a database of the genome, so it's not microRNA specific. So as people find microRNAs in the lab, it may not be annotated to the genome. So remember, these are annotations. It's basically when it's listed on the genome as at this location we have XYZ. Something else just to know is that this whole thing is the gene, the green, but when you have mRNA, this is the coding sequence, CDS. Yellow is CDS and red is mRNA. And you'll generally find more reads, since we're doing mRNA, you'll find more reads mapped to the CDS. So it's common to have these long reads because in the final mRNA product, all of this is spliced out. The fact that we have some reads in the middle isn't concerning. It could be pre-mRNA before splicing. And this here lets me know that it's pre-mRNA. So let me explain this. Okay, the green is the gene, yellow is CDS, red is mRNA. CDS is what's used, coding sequence. It's what's used to make the protein coding, right? The mRNA can be pre-mRNA. So you have this extension here, and then you might also have the middle regions, but for the most part, it overlaps the CDS with extension on either the 5' or 3' end. And we aren't going to worry too much about that, but when you're concerned with microRNA, sometimes you'll have something that maps to the gene. The microRNA will overlay the gene in the reverse direction, and I don't have a good example of that off the top of my head. But these, we can take a look at a different. So sometimes genes overlap and they go, so this gene is going this way, if you can see the arrow pointing this way, and then this gene is going this way. And sometimes those will overlap. That happens a lot in prokaryotes. Practically everything overlaps. In humans, you can see it happening, and sometimes you'll have the microRNA overlapping the gene. Okay, that's not really stuff we have to get into, I don't think. So here's two genes. Here's two genes overlapping. And if I expand this, vertically compressed contact, nope. Oh, here. So if I expand this, you can see one gene here and one, this is a locus, so it's a probable gene that hasn't been assigned a name. One gene here, and then you also have, yeah, they're going two different ways. So this is going this way, and then this locus is going this way. So sometimes you'll have that, and sometimes you'll have the microRNA there. It doesn't really matter. You trust the mapping program. You have to use a mapping program that you do trust, and I really like Genius. I can get into the technicals of why it's good, but I don't want to right now. So nothing maps here anyways. So basically what I have at this point is I have everything mapped for me, and this has microRNA stuff as well as regular genes mixed in. So after mapping, you have what's called a BAM file, and that's basically a list of the sequences and sequence locations that, what am I doing? Hang on.